Hi, I'm David Siegel. I'm here at the Myeloma 2018 meeting with Andrew Spencer and Ravi Vij, and we're here to spend a little bit of time talking about the role of CAR T-cell therapy in multiple myeloma. And I recognize that this is a very complicated question, and perhaps we're asking these questions a little bit early in the history and evolution of myeloma CAR T-cell therapy. But what is your early impression, Ravi, about the efficacy and where you would place CAR T-cell therapy in our armamentarium?
So you just said, I think it's a little early to form a definitive opinion, but most of the studies have been done in highly refractory patients, especially what we call these days penta-refractory patients. I think there's proof of principle that they work. The durability of their working is something that we're just beginning to, I think, understand. Some of the longer-term follow-up data just extends perhaps between a year and two. And I think that certainly the products work, but they probably will not, at least as currently studied in the current population, lead to too many cures, because I think a lot of patients have been looking for the cure for myeloma, and hopefully this will be something that we can tout as a cure in the future with refinement in technology, the patient base. But I think it's certainly an advance, no doubt about it. It's certainly in the population studied, perhaps the most active treatment that we have for our patients. The question also always comes up is, how is the cost going to justify the benefit?
So we were having a conversation about some of this stuff earlier. We have a lot of unanswered questions. Is BCMA the right target? Are there patients who are cured? What percentage of the patient will be cured? How do you see the current technology, mostly directed at BCMA, and where we should go, what kind of questions we should be asking?
Well, I'd like to echo what has just been said. I think that we've got proof of concept that these may be useful in the clinic, and there's certainly a lot of excitement. What I think is probably lacking is knowing the correct target. I'm not convinced BCMA is necessarily the right target. I don't think that the current production process, now the cells are engineered, will probably be the final way that this is done. I think there'll be probably significant advances in the technology. And I think that identifying the patient population in who to use these products will be critical, particularly in jurisdictions where cost may be a greater obstacle to their utilization. And I think really they have to be a home run. I think they have to be a one-off therapy in the right person choosing the right target. So our experience to date has, as you said, been in the unmet need population, what we call the penta-refractory population. And certainly in my own experiences, we've had patients that clearly were not going to respond to anything else and have had exceptionally fast responses that have been durable, but durable meaning a year, sometimes somewhat longer than a year. There's going to be a value decision that needs to be made, and what is the criteria going to be? To cure 25% of patients, to extend progression-free survivals of a year, 2 years, 10 years. What is the answer? I mean, clearly from other CAR T-cell experiences in ALL and lymphomas and in the CAR-19 experience, clearly we are curing a significant portion, and in some diseases, the overwhelming majority of patients. So what is our standard going to have to be? What's going to justify this kind of technology in multiple myeloma?
So I think that's a very valid question, and I think that, I don't know if I have an answer to that question. However, I think it is also going to depend upon the competing landscape. As Andrew mentioned, BCMA may not be the correct target, but if we hypothesize that it is the target worth going after, do you have to have a CAR T-cell? Can you do it with an antibody drug conjugate? Can you do it with the BiTEs in development? I think that will also decide the value proposition for a CAR T-cell. And also I think that finally there will be these novel mechanisms of payment that have started being explored even with the Novartis product in acute lymphoblastic leukemia, but the time point is at 1 month, what is the response, and it's a decision made on payment. But perhaps there may be a model where you can have a phased-in payment that, okay, if you're responding at 1 month, 1 year, 2 years, 5 years, I think those may be solutions to this. I don't know. I'm not an expert on that area, but certainly people are talking about this being a potential way to, answer the question of what the value is.
So, Andrew, you come from Australia and a completely different both philosophy and construct to healthcare in Australia than we have here in the United States. Is there going to be significantly greater barriers? Is this going to be something that is adopted, and what's going to take for it to be adopted?
Well, clearly we have a universal healthcare system, so it's paid for by the taxpayers, and the process to get funding is extremely rigorous. We have to demonstrate incremental cost effectiveness, quality adjusted life year improvement, and there are thresholds for all of these things. So there'll be a major challenge at the current cost to imagine this would ever be funded by the health system in Australia. However, in saying that, I think if the appropriate target population was identified where you could have a reasonably high level of confidence you're going to cure that patient, well, then that would completely change the paradigm. And that's why I think we really need to be doing a lot more work with these agents to focus on who is going to benefit, because we don't really know if they're going to be agnostic to other risk factors and drug resistance mechanisms. So it's really, really early days. There's a lot of hype, a lot of hope, but I think in a system such as the one I'm in, there's going to need to be really rock solid clinical evidence that you are curing people, because it's an incurable disease. So I think if you have a curative intervention, you've got to sit up and really take notice of that.
Particularly if we can utilize this intervention early on in the course of care, avoid the other expenditures. I mean, we're not talking about comparing a extraordinarily expensive therapy with therapy that is of negligible cost. I mean, all of myeloma care has become very, very expensive. So, one can make the argument that if you can, cut out a year of therapy, you can cut out 2 or 3 years of therapy with an early intervention with this kind of technology, that the cost saving is enough to overcome some of our instinctive resistance to buying into CAR T cells as our savior. But I agree with both of you. BCMA is the beginning. We may evolve to other targets. We may evolve to combination targets. I think that's a very active area of investigation right now. But is there anything that either of you would do? Andrew, you first.
Your comment about replacing therapies is actually what drives drug approval where I live. So the funders will look at something and say, well, if I'm going to spend X number of dollars on this, what am I not going to spend money on?
Are Australian dollars worth more or less than American dollars?
Less than American at the moment. And so I would envisage that if we're going to use these therapies, they're going to be used very early in the treatment algorithm. And probably what we'll do generically is get people to minimal disease and then use these agents. Now, how we get there will differ in different jurisdictions. But where I live, it'll probably be something cheap and cheerful and an autograft, and then something like CAR T cells. And I think that could be cost effective as a replacement to other more high cost pharmacotherapeutics.